After C3 cycle, we are going to talk about another cycle that is known as C2 cycle. Now you remember why C3 was C3 cycle, because the first carbon compound that was being formed, it was a 3 carbon compound known as 3-PGA, 3-phosphoglycerate. In this particular process that we are going to study, or rather I would say the cycle that we have the first carbon compound that is being formed is a 2 carbon compound. That is why we are dealing with C2 cycle. But primarily you're not going to come across this name, because the actual name that we come across is photorespiration. Now these are the two people, the duo who was responsible for studying this particular type of physiological process in a tobacco plant in around 1959. What they saw was that, as I told you earlier as well, the most abundant enzyme, that is Rubisco, is little bit deceitful in its activity. Now how it is deceitful? Under the conditions of high temperature and high oxygen concentration, this Rubisco enzyme, which is otherwise conducting the carboxylase activity, all of a sudden becomes more, it has more affinity for oxygen, it develops that affinity and becomes oxygenase in its attitude. Now, altogether what happens is the carboxylase activity is overtaken and instead the enzyme behaves as an oxygenase. Do remember one thing, that this Rubisco, the main active site, is similar for both carbon dioxide and oxygen, but when the concentration of oxygen is high at higher temperatures, especially in tropical plants, it is observed that the carboxylase activity is replaced by the oxygenase activity. Now you remember what happens in the C3 cycle? In the C3 cycle, what happens is that there is a carbon dioxide molecule. It comes and joins with RUBP. Where there is RUBP, there would be Rubisco. Now this RUBP is a 5 carbon compound. What happens in the case of photorespiration? If we simply see its definition, it says that photorespiration is a process that leads to evolution of carbon dioxide. Okay. Now, when we talk about photosynthesis, we want that carbon dioxide should be absorbed and it should be assimilated in glucose. Now, in this process, it is happening opposite. The carbon dioxide is being released. So, first thing that you have to remember, that this is a totally wasteful process, not in the interest of plants. Why it is not in the interest of plants? Because if so many reactions are taking place, as you can see, I have mentioned over here, there would be some sort of energy which would be required. Energy is not being generated, energy is being utilized, and instead, even after utilizing the energy, the carbon dioxide is not getting fixed, it is getting released. So it is a wasteful process for the plant. The only benefit that photorespiration has for the plants is that it avoids or it restricts the photooxidation at higher temperatures. And the tropical plants, they suffer the maximum brunt of this particular reaction, that is photorespiration, where the carbon dioxide which was otherwise to be fixed into the plant, that is getting evolved. So there is no particular indicator for photorespiration as such, but one can make it out that plant is not performing enough photosynthesis. So when the rate of photosynthesis is low, then the photorespiration rate is higher, and when the evolution of carbon dioxide is higher than expected as from the plant, then also we can make it out that it is undergoing photorespiration. Now what is this photorespiration all about? As I told you, there is the compound known as RUBP. In the presence of Rubisco and carbon dioxide, it was forming 2 molecules of 3-phosphoglycerate. That was what was happening. Now what will happen? RUBP in the presence of Rubisco, in the presence of oxygen, which is higher than carbon dioxide. Oxygen and carbon dioxide both are present in the air, but the oxygen concentration is higher, which is automatically higher. We all know that oxygen is 23% of the atmosphere, while carbon dioxide is not even 1% completely. But at higher temperatures, this concentration of oxygen is favored by Rubisco, and the activity which comes at place of carboxylase activity is that of oxygenase. And when oxygenase acts, what it does is it converts this 5 carbon compound, instead of converting it into 2 3 carbon compounds, there is formation of 1 3 carbon compound and 1 2 carbon compound. This is what is formed, okay? And once the outcome is not similar to as seen in C3, there would be totally different course of events that would take place. So we know that 3 carbon compound is 3 PGA, 3 phosphoglycerate. So one molecule of 3 phosphoglycerate is formed that goes to C3. Okay, that would be utilized in C3 for whatever use it would be. It should need 2, but there is 1 PGA that is formed, that is 3 carbon compound is formed. It goes to C3 cycle, and the 2 carbon compound, that is 2 phosphoglycerate, glycolate, sorry, not glycerate, 2 phosphoglycolate enters the photorespiration cycle. You have to remember that photorespiration is also a cyclic process. That is why I said that it is known as C2 cycle. What has happened in the beginning? The point of difference is that RUBP is there, Rubisco is there. Only thing that at higher temperature, oxygenase activity is higher. So what happens is, instead of forming 2 3-phosphoglyceric acid molecules, it forms 1 3-phosphoglyceric acid molecule, and one molecule that is formed is 2-phosphoglycolate. Now this reaction is taking place in chloroplast. This photorespiration takes place in 3 different organelles, cell organelles. First is chloroplast, second is peroxisome, and third one is mitochondria. Now as you can see, the first reaction where this Rubisco was available, that has to take place in chloroplast. In chloroplast, the phosphoglycolate that is being formed, it combines with water and forms another compound, that is glycolate. Now this glycolate would move to peroxisome. Remember that in peroxisome there would be deamination and amination. So first of all, we are going to see that how this glycolate, first of all it gets oxidized and forms glyoxylate and peroxide. Later on, this glyoxylate gets deaminated and it forms glycine. These are the steps. See, first step is that the phosphoglycolate that was formed by activity of Rubisco in the oxygenase advent, it forms glycolate by combining with water in the presence of phosphatase enzyme. This glycolate gets converted to glyoxylate in the presence of oxygen. This reaction of the formation of glycolate takes place within the chloroplast. Rest reactions are taking place in peroxisome. Now this peroxisome takes up the glycolate. In the peroxisome, there is a process where the glycolate takes up oxygen. It forms glyoxylate. In parallel, this peroxide which is formed, that gets converted into water and oxygen, and there is generation of more oxygen. Whatever is being used, that has been regenerated. The glyoxylate undergoes deamination and forms glycine. Now this glycine is an amino acid, okay. Now 2 molecules of glycine, these glycine would get transferred to mitochondria. 2 molecules of glycine along with NADP positive, they would undergo decarboxylation in the presence of decarboxylase and form single molecule of serine, that is again an amino acid, and there would be release of carbon dioxide, ammonia and NADH. This is the step that takes place within mitochondria. Now the serine which is being formed, it goes back to peroxisome. As you can see over here, alpha ketoglutarate was a product of reaction where glycine was formed in the peroxisome. The same alpha ketoglutarate, when serine moves back to peroxisome, combines with it and it forms hydroxypyruvate and glutamate is formed. Okay, the same glutamate that was present in the mitochondria, the same peroxisome. Now this hydroxypyruvate would combine with NADH. Over here there would be amination and glycerate would be formed. This glycerate on combining with ATP would form 3-phosphoglycerate, the similar one that was used for carboxylation. This 3-phosphoglycerate would combine with the 2 carbon and form RUBP. Okay, so this is all these are all the steps that take place in photorespiration. For now, you have to keep these in mind, you have to learn them, that these are the steps which are taking place. What is to be kept in mind in particular is that instead of taking the pathway that was responsible for formation of glucose, here the carbon dioxide is getting released, ATP are being used, but we are not getting glucose. So this is a wasteful process. The 3 different organelles where this process takes place, that is to be remembered. And before you jump to the next lesson where the cyclic representation of C2 is being discussed, you just keep in mind that these reactions, if they are learned by heart, it would be easy for you to understand that particular lesson. And lastly, this is a wasteful process and we have to study it, unfortunately.